faithfully delivering value and interpreting great books from around the world. Hi, this is Sally's Book Talk. Today, I'll be discussing the book Hemingway's Cat Legend for you. The literary giant Hemingway once owned a cat that was just as famous as he was, not because it was a great mouser, but because it had 6 toes. Whenever visitors tour Hemingway's home, guides always highlight the 6 toed cat and call it Hemingway's cat. Actually, it's like something straight out of a Hemingway story. Oh, this rumor is totally made up. Hemingway didn't own cats on Key West. His wife preferred peacocks, but that doesn't stop the 6 toed cats. So, alongside Hemingway's fame, do 6 toed cats really exist? Yes, they do exist. So, why do 6 toed cats happen? This is precisely Hemingway's cat. This book aims to solve that mystery. The subtitle of the book is How Do Genes Work? So, clearly, the 6 toed cat has a genetic issue. The author, Kat Arney, is a genetics PhD from Cambridge, now a freelance science writer and BBC host, dedicated to popularizing science. Using the 6 toed cat as a hook, the book dives us into the tiny world of genes to explore how they function. So, first up, we need to tackle what exactly is a gene? Surprisingly, the book shows that there's no official definition for gene. It's just a handy term, but its meaning is pretty fuzzy. After looking at various scholars' takes, the author offers her own definition. A gene is functional genetic material, like DNA and RNA. DNA stands for deoxyribonucleic acid, a big molecule in the cell's nucleus. Its structure is like a twisted zipper, the famous double helix. On this zipper, each tooth matches up. Their chemical structure is called bases. With only 4 types, how do they encode all that complex genetic info? The answer's in the order. At the same DNA spot, yours might be ATCGAA, mine might be ATCGCA, and that decides our different features. And RNA, short for ribonucleic acid, acts like a messenger, translating DNA into proteins. That's why it's called messenger RNA. Gene function works through DNA and RNA interactions. DNA gets transcribed into messenger RNA, which then gets translated into proteins. This is what scientists call the central dogma. They generally believe they understand gene function well. Genes strictly follow the central dogma. But as research advanced, many new gene operation patterns have emerged. The author introduces several key gene strategies. Genes can self-edit without human interference, showing powerful editing capabilities, like cutting, pasting, modifying, even deleting themselves. Plus methods still hard to grasp, like gene jumping interference, making once simple DNA sequences complex and profound. Through these strategies, we glimpse fundamental principles of gene operation. First, the weirdest strategy, the junk strategy. The junk strategy means packing functional genes with seemingly useless junk genes, like in a big factory where only a few actually work, while thousands just pretend. Few would treasure garbage. But junk genes are the exception. They're seriously called junk genes, but they're actually proper DNA sequences. This name probably came from frustrated researchers. They couldn't figure out these genes' purpose, so just called them junk. Problem is, junk genes are everywhere and super abundant. How abundant? Some say over 90% of human genes are junk. We have similar gene counts to pufferfish, but 8 times more DNA. Yet pufferfish thrive and even produce deadly toxins, showing extra genes aren't necessary, likely all junk. Despite humans having 90% junk genes, we're not the species with the most. Onions have 5 times more DNA than humans, and there's a plant called Paris japonica that actually has over 100 times more DNA than us. They're basically junk gene marketplaces. It's like an old house lived in for years. The longer you stay, the more junk accumulates. Junk genes are essentially the accumulated trash from long-term evolution. Without cleanup, the longer evolution continues, the more junk genes pile up. Biological systems result from millions of years of evolution, accumulating random genetic fragments. Evolution isn't precise, but rather good enough. Natural selection turns a blind eye to junk genes unless they affect survival. Otherwise, everyone just muddles through. To draw an analogy, genes are like bumbling bureaucrats who sometimes complicate simple things. Though inefficient and costly, genes happily accept this bloated state. Thus, genomes aren't precise blueprints, but ever-changing drafts full of junk. A genome refers to an individual's complete DNA sequence containing all genetic information. Now, you might wonder, are junk genes actually useful? Some scientists believe they must be useful, we just don't know how yet. Like old clothes in a corner, someday you might make rags or a mop from them. Same with junk genes. Useless now doesn't mean forever. They might prove valuable someday. These seemingly functionless genes are like foam padding in delivery boxes. Not what we ordered, but essential. They provide crucial protection. Junk genes are actually unsung heroes protecting functional genes. This view gains increasing support, and we'll soon see evidence backing junk genes. We've discovered some junk genes have specific regulatory functions. Like in that factory analogy, some don't work but direct others what to do and when. Now, let's discuss the second major strategy of gene operation. The regulation strategy. Just like train operations need control, genes also require regulatory execution. Regulatory genes act like light switches, controlling gene activity. Some enhance it, others weaken or even shut it down. In classical molecular biology, genes primarily direct protein synthesis, the one gene, one protein principle. We later discovered some genes don't make proteins but specialize in gene regulation, regulatory genes. The regulatory genes play crucial roles in biological development. For example, we all start as a single fertilized egg that develops into diverse cells, heart, liver, nerve cells, each with unique functions. Their differences emerge through regulatory genes altering gene activity, turning some on, others off. Essentially, gene regulation is simple. Specific proteins bind regulatory genes to activate or deactivate genes at precise times or locations, creating diversity from identical genomes. When regulatory genes combine, their effects amplify dramatically. Single regulation is like opening one door to a single room, but combinatorial regulation lets you open multiple doors, each leading to different paths. Macroscopically, gene regulation is evolution's key. Humans and chimps look completely different. Previously, we thought genes cause major species differences, but humans and chimps share over 99% identical genes. We became human while chimps remain chimps primarily because regulatory genes make the difference. Their cumulative effects build variations that assemble identical genes into completely different cells and bodies. So that's why it only takes about 20,000 genes to assemble a human. The regulatory genes are so crucial. If anything goes wrong, the results can be unpredictable, like with Hemingway's cat, where gene regulation failed. Scientists discovered an intriguing gene in fruit flies, later found in other animals too, the Sonic Hedgehog gene. Hedgehog gene, this quirky name comes from the Sonic the Hedgehog video game. The hedgehog gene's job is guiding embryos to grow fingers and toes in the right places at the right time. But this gene rarely messes up since it's controlled by another regulatory gene. Though 800,000 bases apart, the regulatory gene remotely controls the Sonic Hedgehog gene. And if just one base error in the regulatory gene changes Sonic Hedgehog's activity, it makes normal cats grow 6 toes, creating Hemingway's cat. A tiny tweak affects everything. That's gene regulation's power. That's exactly why scientists are digging deeper into gene regulation research. As research advanced, a third gene strategy quietly emerged: methylation. A methylation strategy adds a methyl group to DNA. This process is called methylation. Simply put, a methyl group is a tiny molecule, 1 carbon, 3 hydrogens, and methylation can alter a gene's entire activity. Imagine building blocks. If you glue a tiny plastic piece to one block, it changes how all blocks fit together, reshaping the whole structure. The methyl group is like that plastic piece. If someone inherits their father's gene sequence, but with one extra base added, the genetic information changes. Though tiny, it can sway an organism's entire fate, and this fate can cross generations. Thus, methylation instantly became a research hotspot, but media overhype created many misconceptions. Mass media loves hyping methylation's mystical effects, using clickbait headlines for ambiguous studies, especially about cancer links. Oh, methylation seems more obvious in tumor cells. Some science illiterate journalists confidently claim methylation research can cure cancer, but scientists aren't nearly that sure. To scientists, no clear evidence links methylation to cancer. Claims that environments cause methylated genes to pass to offspring are also unreliable. Mostly, it's media exaggeration. What we know, methylation regulates genes, but its exact functions need tons more research. Still, methylation's crucial. It's everywhere, super sensitive, and even affects gene editing. Speaking of gene editing, that brings us to the fourth strategy, splicing. Gene splicing is basically like editing documents, cut some text, paste others together to form new sequences, new genes. From roses to elephants, fruit flies to humans, cells are splicing genes like crazy. Splicing targets messenger RNA. After transcription from DNA, it's semi-finished, chopped into sections, some discarded. The kept parts? Exons. They get reassembled. How do exons splice together? Say a gene transcribes to mRNA, gets cut into 5 segments. Toss 2, keep 3. Those 3 can combine freely. Segment 1 plus 2, 2 plus 3, or wilder combos, virtually no restrictions. Through endless splicing, they translate into multiple proteins. Now we see why few genes produce tons of RNA, like film editing. Same raw footage yields different movies. Longer reels, more complex possibilities. 3 exons are just a simple demo. In reality, it's way more complex. For example, one fruit fly brain gene splices into 38 exons, forming over 30,000 mRNA variants. Human brain cells, hundreds of such genes, each making hundreds of RNA versions. Like investing $1 and getting tens of thousands in return. RNA splicing is essentially massive gene editing with huge returns. Cutting whole sections, deleting large chunks, broad strokes with strong impact. Besides this, cells make fine microadjustments to RNA, changing individual letters after DNA transcription without altering the DNA sequence itself. So from one DNA set, we get varied RNA. These tweaks are super common. In the human cerebral cortex, nearly all messenger RNA in neurons gets edited. In the cerebellum, about half gets edited. This complex editing lets few DNA sequences produce tons of RNA variations, creating protein diversity from once monotonous genes. That's how humans build intricate bodies with just 20,000 or so genes. Beyond cutting and pasting, genes need a fifth strategy, RNA interference. RNA acts like a mail carrier, delivering genetic info from DNA to proteins. But sometimes this carrier tampers with the mail. That's RNA interference. So what exactly is this phenomenon, and how was it discovered? Researchers used morning glories. They inserted a purple pigment gene in the blue morning glories, creating a new hybrid. They expected deep purple flowers, but the results shocked everyone. Not only no deep purple, they lost the original blue too. The new blooms were white. Later studies showed extra genes produced extra RNA that silenced normal genes. This RNA interference phenomenon, where added genes created RNA that blocked pigment genes, accidentally made white flowers. RNAi works across all life forms, and the dosage is tiny, just a few molecules per cell, yet the effects are remarkable. A single RNA molecule can shut down a gene and affect the whole body. Whether introduced into nose or tail cells, it creates systemic effects. This immediately made pharma companies consider RNA interference for new drug development. Sure enough, just 6 years after RNAi's discovery, a related drug entered clinical trials. Today, RNAi-based drugs treat hepatitis B and leukemia. RNA interference isn't just a genetic strategy. It's become humanity's health-protecting weapon. But one question has long puzzled scientists. From a gene operation perspective, why do cells need RNA interference? What purpose could it serve for cells? RNAi has practical value. It might be cells' ultimate virus fighting trick. Many viruses synthesize RNA from their own DNA after invading cells. TILs detecting foreign RNA go haywire, shutting down gene activity to counter the threat. This evolved into RNAi helping cells turn genes on or off at precise times and locations. Now, microRNA is rapidly becoming a research hotspot. In a way, microRNA is a form of RNA interference. These special small RNAs, present in animals and plants, participate in lifelong gene regulation. Finally, the sixth gene strategy, jumping strategy. Jumping genes hop around genomes, giving cells new traits. All organisms have jumping genes, humans included, mostly boring repetitive sequences. One ancient human jumping gene, just thousands of letters, copies itself over 500,000 times. Like a virus, it tricks cells into copying itself, cuts random genome gaps, jumps in and integrates, adding copies that sometimes confer advantages. For instance, some leaf monkeys unexpectedly gained HIV resistance thanks to jumping genes. But mostly gene jumping causes harm or outright cell collapse. Random jumping and inserting is dangerous. It can wreck original gene sequences. Now reconsider junk genes, you'll discover another hidden value. Since junk genes are trash, they don't mind random insertions. They stay junk anyway, right? Thus, abundant junk genes let genomes resist jumping gene damage. This junk gene behavior really resembles foam padding in delivery boxes. Beyond junk genes, cells developed other ways to stop jumping genes. Like methylating jumping genes. Once methylated, they can't jump. Many jumping genes get locked this way, minimizing harm. Overall, though, jumping genes do more harm than good, mostly damaging or useless. But new research rebrands jumping genes, revealing fresh value. Earlier, we said adult animals methylate most jumping genes, silencing them. Yet in mouse neurons, researchers found activated jumping genes. They repair cells, boost memory, and enhance brain processing. Surprisingly, elderly human brains show more jumping gene activity, causing two outcomes. On one hand, improved brain processing and anti-aging. On the other, accumulating genetic damage leading to Alzheimer's. This dual nature applies to all gene strategies, helpful yet with side effects. Genes carefully weigh these trade-offs. Maximizing benefits while minimizing harm is their ultimate strategy. Gene operations extend far beyond these six strategies. At the genetic level, mysteries remain, like zombie gene resurrection and RNA circularization. Scientists can observe phenomena but don't know why they occur. But hey, temporary confusion is just part of scientific progress. Ultimately, this book reveals gene operations' profound complexity. It's this unpredictability that breathes vitality into life. This mystery also warns us. Don't recklessly tamper with genes. Their power, wow, exceeds our imagination. Amidst enigmatic destinies, genes and environment forge unique beings. Such extraordinary existence deserves celebration and cherishing. All right, that concludes our journey through Hemingway's Cat. Reading notes are below the audio. See you tomorrow.